Okay, ladies and gentlemen, next agenda is opening remark from Head of Research Center for Applied Microbiology BRIN to Dr. Ahmad Fatoni. Time is yours.
Yang bocor di close dulu. Oke. Bismillahirrahmanirrahim. Thank you Mbak Puji as our MC today, Honorable Speaker Prof. Sukuri from MTech University Putra Malaysia. Respected partisipan, Bapak Ibu semuanya, para profesor, para periset di lingkungan ORHL maupun dari BRIN yang lainnya. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera, sehat bagi kita semuanya. Very good morning everyone. First of all, I would like to thanks to Prof. Suguri for accepting our invitation today, as actually he's here just next to me. Because today webinar we run hybrid, so we have here offline about 20 participants and in online about 100 participants. And welcome Prof. Sukuri to Cipino. So Prof. Sukuri is here with us as visiting researcher. Started 2 weeks ago. Yeah, 2 weeks ago. Yeah, okay. And we are happy to have Prof. Sukuri and actually his partner and his his wife as well. Yeah, his partner with us together today. Yang kedua, saya ucapkan banyak terima kasih kepada Bapak Ibu semuanya, yang teman-teman yang sudah hadir di sini di Luring di PNC, kemudian juga seluruh para periset yang hadir di online. Jadi kita hari ini akan mendiskusikan atau melihat nanti ini topik yang mungkin baru buat saya yang pasti, dan mungkin sebagian juga Bapak Ibu, dan nanti Bapak Ibu yang juga sudah familiar kita akan perkuat lagi. Tetapi webinar pada pagi hari ini untuk persiapan kita nanti akan segera launching platform kolaborasi biologi struktur. Jadi so please allow me to share one slide, Prof. Sukuri, before we have a presentation. So please let me. Sorry, share slide. Udah belum ya share-nya ya? Oke. Bapak Ibu sekalian, jadi ini sekalian informasi juga di awal ya Bapak Ibu, bahwa nanti kita sesuai dengan amanat dan juga yang sudah disampaikan Pak Kepala BRIN juga beberapa waktu lalu atau selalu di beberapa kesempatan, bahwa BRIN kita akan ada salah satu platform kolaborasi yang namanya platform kolaborasi biologi struktur biomolekul keanekaragaman hayati. Di Zoom sudah selesai belum ya?
Sudah.
Slide. Oh, yang itu tampilan Zoom ya?
Recording in progress.
Bentar. Izin ya Bapak Ibu ya, sebentar. Kendala di share screen. Belum ya. Hmm. Oke, baik Bapak Ibu. Jadi untuk informasi dahulu di awal, jadi ini memang belum secara ofisial dirilis, karena nanti kita masih dalam rangka penyusunan proposal saat ini, kita akan kirimkan ke DPRI, kemudian nanti kalau sudah nanti disetujui, baru kita akan lakukan open call. Hanya saja terutama kemarin di internal ya, kita sudah coba untuk exercise dengan open call for ICP, dan kita kemarin sudah dapat 70 ICP juga untuk melihat seperti apa nanti potensi kita ketika kita akan lakukan open call for collaboration. Jadi namanya nanti platform kolaborasi untuk biologi struktur biomolekul keanekaragaman hayati. Nah, nggak bisa ya? Oke. Nah, agak lambat ya koneksinya ya. Sorry about the connection. Ya, jadi Bapak Ibu, nanti untuk platform kolaborasi yang akan kita open nanti, ini soalnya desain kok belum ada ya, beda ya. Jadi nanti kalau di bagian paling kiri itu, oh ini karena ini share-nya Mas Andi ya, oh di sini ya. Oh oke, pantesan beda ya. Oke. Jadi nanti Bapak Ibu ya, kita nanti asal resources-nya, KHT-nya itu kita akan mencakup semuanya, mau dari plant, animal, maupun microbes ya Bapak Ibu. Kemudian yang akan nanti kita proses sampelnya itu dalam bentuk ada 3 ini Bapak Ibu skopnya. Dia dalam bentuk protein, from animal, microbes, and plant. Kemudian kompleksnya di sini adalah protein berasosiasi dengan lipid makromolekul ya Bapak Ibu ya, lipid, karbohidrat, asam nukleat, kemudian juga sel dan jaringan. Jadi prinsip nanti Bapak Ibu ketika mau meng-apply terkait dengan biologi struktur ini, patokannya ke sini gitu ya. Sampel kita adalah protein, kompleksnya, dan sel dan jaringan. Kemudian nanti kita akan proses menggunakan toolnya adalah utama KWM, dan output utamanya di sini adalah struktur 3 dimensi. Jadi itu yang menjadi patokan nanti Bapak Ibu dalam menyusun ICP ataupun proposal nanti. Output utamanya adalah struktur 3 dimensi dari protein maupun kompleks sampel Bapak Ibu semuanya. Karena nanti dari struktur itu adalah from structure utamanya nanti untuk studi fungsi dan interaksi. Jadi itulah yang menjadi ruang lingkup dari platform ini. Gitu. Kita tidak nanti bicara untuk development vaksin contohnya atau apa enggak, tapi kita fokuskan ruang lingkup adalah bagaimana kita bisa mengelucidasi 3D structure, kemudian dari structure itu kita bisa tahu shape, fungsi, dan juga interaksi kalau itu dengan antar biomolekul lainnya. Nah inilah yang ruang lingkup utamanya Bapak Ibu ya. Kemudian next-nya dari itu ya. Oke, next berarti. Bentar, nggak bisa ya. Oke. Ini roadmap saja Bapak Ibu ya, selama 3 tahun nanti pendanaan akan kita lakukan selama 3 tahun Bapak Ibu semuanya, dengan outputnya tadi kita sampaikan struktur 3 dimensi, kemudian publikasi, tambahannya ya publikasi, kemudian KI ya, KI, lalu juga di sini yang menjadi pembeda nanti adalah ada SDM-nya juga Bapak Ibu unggul, DPR kita nanti. Jadi platform kolaborasi itu mendapatkan sedikit privilege ya Bapak Ibu. Jadi nanti satu periset itu kita bisa, atau kita akan encourage teman-teman nanti yang akan terlibat langsung harus melakukan pembimbingan juga gitu ya, DPR 1 S2, 1 S3 gitu. Jadi nanti bayangan kita kalau satu periset itu bisa membimbing 2, 1 S2, 1 S3, itu kita akan setiap tahun minimal 10 DPR ya gitu. Nah itu nanti privilege-nya itu untuk H-index bimbing kita nggak akan dilihat gitu ya. Kita akan buat panduan khusus, karena ini kita dari Pak Kepala menyampaikan kalau platform ini tujuan utamanya malah kita harus meng-create new talent yang memang bidangnya biologi struktur, karena existing kita sangat masih sangat rendah atau masih sangat kurang, sehingga untuk akselerasi itu silahkan nanti Bapak Ibu yang nanti mendapat pendanaan ini cari dari sekarang kandidat S2 S3 yang nanti fokusnya di biologi struktur gitu. Nah, nanti teman-teman yang terlibat di PR nanti langsung kita usulkan juga ke RA, jadi riset asisten. Jadi ini juga salah satu privilege juga ya. Jadi harapan kami bisa jadi motivasi juga bagi teman-teman untuk nanti bantu risetnya itu bisa nyari mahasiswa itu. Jadi mahasiswa nanti jadi DPR dan DPR nanti dia juga dapat RA. RA kita nggak perlu submit tempat luaran gitu ya, karena ini adalah kita apa namanya top down dan memang tujuan utama itu bagaimana kita bisa lebih cepat lagi untuk meng-create talenta muda. Jadi nanti mohon Bapak Ibu bisa mulai hunting ya ke kampus-kampus itu untuk yang bidang biologi struktur ini. Kemudian selanjutnya yang terakhir, sorry for long opening, Prof. Syukur, because I need to explain at first. So Bapak Ibu, jadi kalau kita lihat di sini adalah strategi kita pelaksanaan di platform kolaborasi ini, Bapak. Jadi main faktornya, basisnya itu memang dari kolaborasi. Jadi kemitraan, terutama yang sebelah kiri itu adalah dari luar negeri terutama Bapak ya. Jadi seperti yang saat ini kita lakukan, kita ada Prof. Syukuri dari UPM dan beliau memang bidangnya biologi struktur di MTech UPM, dan nanti dari situlah kita kemudian kita bisa kembangkan joint risetnya. Jadi kita nanti akan diskusi topik dengan Prof. Syukuri, kemudian kita usulkan gitu ya. Itu nanti kemudian kita akan lakukan open call, lalu kalau pendanaan nanti dari DFRI ya, dari yang sebelah kanan itu Bapak panahnya ya. Nah, nanti akan ada 3 tema, Bapak Ibu, 3 tema, protein, kompleksnya, dan sel jaringan. Nanti ada subtemanya juga yang nanti kita akan detailkan. Nanti semuanya akan dilakukan secara website. Kita sedang siapkan juga platformnya. Kemudian nanti untuk strategi outputnya KTIKI itu tentunya dengan basis kemitraan untuk mendukung KTIKI luarannya. Kemudian kita juga akan maksimalkan postdoc, VR, DPR, dan RA ya, yang nanti untuk mendukung luaran KTI kita. Kemudian untuk SDM unggul, kita selain dari mobilitas preset, kita juga akan lakukan workshop dan seminar, termasuk hari ini adalah rangkaiannya. Pertama kita run untuk webinar series on structural biology, bagaimana kita open dulu, kita perkaya dulu insight terkait dengan biologi struktur, baru nanti ada chance in paralel kita nanti submit proposal, Bapak Ibu bisa lebih tahu insight terkait dengan biologi struktur, bisa menyiapkan atau mempertajam ICP yang kemarin sudah dimasukkan, atau mau mengusulkan baru silahkan, kita masih open ya nanti. Dengan adanya jadi kita coba running setiap minggu sekali untuk upgrading atau webinar terkait dengan biologi struktur dengan mengundang narasumber. Nanti mungkin kita dari Jepang ada, kemudian dari Finland, dan dari mana kemarin ya. Ada beberapa negara dari luar negeri nanti kita coba undang sebagai narasumber. Itu Bapak Ibu ya, ini sekilas saja terkait dengan apa yang nanti insya Allah kita coba running di tahun ini dengan tentunya dukungan Bapak Ibu semuanya. Semoga platform kolaborasi ini benar-benar bisa menjadi lompatan Bapak Ibu ya, riset di kita terutama di biologi struktur, karena basis utama kita adalah di bagaimana kita tahu struktur dari sebuah makromolekul ini. Karena di sana nanti Bapak Ibu mau mengembangkan advance, mau ke arah protein engineering atau mau ke arah rekayasa sintetik biologi lain-lainnya, tuh basisnya semuanya kita harus tahu dulu strukturnya itu seperti apa gitu ya, yang akan kita otak-atik atau kita akan modifikasi. Jadi di sini adalah menjadi pintu gerbangnya. Nanti setelah ini Bapak Ibu bisa advance lagi mendevelop produk maupun mendevelop riset Bapak Ibu berbasis setelah kita tahu dari struktur dari apa yang kita kerjakan. Mungkin itu Bapak Ibu semuanya ya. This is all that I can say about very brief introduction about structural biology platform that we are going to run this year. But please be patient. We are going to submit proposal first, and then we will inform when open call for collaboration is open ya. So silakan Bapak Ibu, nanti kita simak sama-sama dari narasumber utama kita Prof. Syukuri hari ini diskusi, terutama yang luring nanti bisa untuk mempertajam Bapak Ibu ICP kemarin atau mau mengusulkan baru, very welcome ya Bapak Ibu ya. Jadi dari 70 itu harapannya nanti kita bisa nambah minimal 100 gitu ya. Jadi dari 100 kan nanti masih diseleksi ke proposal. Jadi reviewer kita juga sudah ada, dan silahkan nanti Bapak Ibu terutama coba kalau ada kalau kolaborasi dari luar negeri itu bisa discuss terkait dengan riset ini. Mungkin itu dari saya. Terima kasih Bapak Ibu semuanya yang telah hadir. Semoga nanti kita bisa diskusi lebih banyak lagi. Thank you Prof. Sukuri for later on you give a presentation about biology structure. So by saying Bismillahirrahmanirrahim, I officially open this seminar. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih Bapak Fatoni atas sambutannya. So before we start the next discussion, we will have a photo session first. To all participants, please activate your camera and for the committee to take the screenshot. Okay, I will start count from first slide. Ready? 1, 2, 3. Next slide. 1, 2, 3. The last slide. 1, 2, 3. Okay, thank you. Ladies and gentlemen, now we are going to start our discussion for today, which will be led by Vina Amrita Laksmi, researcher from Research Center for Applied Microbiology, BRAIN. So, Mrs. Vina, time is yours.
Thank you very much for the MC. Sorry. Gimana sih cara nyalainnya, Mas? Hah? Oh. Okay, thank you very much. So today we are honoring to welcoming Professor Muhammad Sukuri from University Putra Malaysia. So I would like to briefly introduce him to our honorable colleagues here. So Professor Sukuri is head of Enzyme and Microbial Technology Research Centers until now. And then despite his very young age, he has very great achievement under his belt. So now he is part of the expertise in the Malaysian Society for Biochemistry and Molecular Biology, Malaysian Society for Microbiology, Malaysia Crystallography Association, and also Malaysian Protein Science Society as a founder. And even in the international level, he is a member of the Asia Pacific Protein Association Council member. And then also a frequent user in the Super Photon Ring 8, Spring 8 in Japan. Also member of the Protein Society of USA, member of the Biochemical Society in United Kingdom, UK, and also member of the European Society of Applied Biocatalysis. So now we are very excited to listening to his transfer knowledge to us. So I hope we can get many things and learn many things from him as well. So welcome, Prof. Sukuri. So yeah, time is yours, Prof. Now. Yes.
Yes, please.
Assalamualaikum warahmatullahi wabarakatuh. We all here to say online. We did all here. Suppose. Okay. So itu dia. Terima kasih atas dukungan kemudian di pasar dan di forum ini. Terima kasih kepada Pak Tony dan juga Center for Applied Biology. Dan untuk saya untuk berisi sebulan di Indonesia, bersama tulis di all biology foods and penting di sini saya dapat fungsi dan share share about learn students, the team, and both the collaboration shall see and worldwide. Jadi, let me talk a bit in English. So basically, what I heard from Tony, we use for the more one structural value approaches. So I think probably my talk today try to just so I can try to give you some ideas a bit about what is a bit about structural biology in general and how is structural biology are being used in current tools nowadays to understand many aspects of the right.
Okay, so right. Sorry for the inconvenience. Let me just proceed. Okay, to my talks will be more on introductory of a bit on structural biology. We cannot go too deep about this one. I try to be a bit open, and then follow through is about some of the story, the outline of our success stories in our laboratories, and perhaps from there, the audience and listeners should be able to have some ideas about the direction and what this those ultimate tools should be able to give us in way forward. Right. My name is Muhammad Shukri Umar Ali. So I'm from. You can just people call me Shukri lah. My name can be very long, Muhammad Ali. So people call me Prof. Shuk lah, Shuk in short. So I'm heading the Enzyme and Microbial Technology Research Center. In short, we call it MTech. One of the we call this Research Center of Excellence in University Putra Malaysia. So the affiliation of Faculty of Biotechnology and Biomedical Sciences UPM. Okay, so let me just go through a bit about what is before I start. Showing perhaps all of us should know at this moment the three-dimensional structures of proteins. Okay, so this is basically an enzymes or lipase that performs the hydrolysis and the degradations of lipid in general, and it can also perform wonders where this enzyme can perform reverse reactions, where it is not commonly known for other enzymatic processes. So by understanding its structure, for example, how the protein folds, how the protein activates and how the enzyme function, we should be able to get some good idea about how this enzyme works. Okay, so I will go into detail about this later on. There's even a protein that looks like a question mark. Okay, so more and more questions to be asked later on when it comes to this particular topic. So basically, in general, this is not a new tool, a new approach, a new field. It's been long before. I think since the discovery of X-rays by Röntgen back in 1914. So this crystallography, for example, one of the tools in structural biology has been around for more than 100 years. Okay, but then again, as time moves by, more and more advanced, more techniques has been developed, which is now the cryo-EM is becoming more emerging techniques and in assisting the structural biology and give us the shapes, give us the understanding about the structural functions of biological macromolecules. So from the structure, basically, whenever researchers try to delve and trying to get into this structural biology aspect, we want to the key point is what we want to see from our given structures. Okay, so it's not that we solve structures for fun, to just keeping a logbook. We want to see what is it afterwards in the structure that we obtain. That could actually decide on what are the methods and approach that we can actually employ. But of course, throughout the process, in this approach, we should be able to understand in terms of biomolecular level of understanding in terms of shapes, sizes, and of course, what is important is the being able to see things using tools that we have that I'll be listing a bit later on at the atomic level. atomic level. So from there, people have been able to coin terms like the terms called picobiology, looking into the processes of understanding of electrons and interactions of metal at the atomic level. So, and nowadays structural biology has expanded to certain fields, not just in terms of biology. There are also structural microbiology, structural enzymology, structural chemistry. So it expands. So people can come up with your own specialty when we employ this structural processes are being employed. We can, of course, I'm more into structural enzymology, looking to the aspect of not just the classical enzymology process, but also into the which to understand mechanism, enzyme mechanistic at the atomic level. Okay, so yeah, so it is very important. I hope that of course, it's just that we are not just being fundamental about this particular aspect, but of course, when the term when the term structural biology is coined, so we thought that it is more into fundamental aspect, but basically it is not to that aspect actually. So we can actually include what the topic that to be studied, for example, can be useful in new drugs development. I'm sure that we all know that when during the post-pandemic era, so during the pandemic, we all know very well about COVID and how the so-called the capsid, the protein, and the coronavirus has been very much involved in the infection. So actually, that is with the assistance of structural biology tools, structural biologists and biochemists. So to give information to pharmacists, to I call engineers to produce this protein of interest and eventually to produce vaccines and now come up with fragments of RNAs to be, of course, included to be put in our body as vaccines from Pfizer, for example, and AstraZeneca. So from the understanding of the structure to the CRIU-M methods and of course structural X-ray crystallography, this has actually expedited the process. Okay, so and within 1 year, vaccine has been able to develop. This is immensely very fast actually. So of course we should be able to understand as far as the scope is concerned, understanding disease mechanism, right? And from there we also, most of which I'm sure a lot of us, when we employ structural biology, we want to study proteins, how protein works, okay, how these so-called enzymes or antibodies or many which plays important role in many cellular processes, right? So of course, in the case of enzymes, we are looking into improvement of enzyme activity. So how do we, which part of the enzyme that is called important or detrimental for any given reaction processes? Okay? And of course, to the larger extent, to the larger scale of understanding is to unravel the process of our fundamental biological processes. I think what has been outlined by Dr. Fatoni before, certain level of protein understanding from the protein, subunit, multiple subunit, and up to the cellular level. So this could give an idea about many aspects of understanding of life by the use of structural biology. So in our case, for example, in my case, so we are really interested in about enzymes. So us, not just by trying to get enzymes from the microorganism, but of course, what are we going to do with the enzymes? So nowadays, industrial applications requires enzyme to be able to withstand in many harsh processes. Enzyme to be able in manage solvent, the highs and the lowest temperatures, the extreme pH. So natural enzymes may not be able to do that. Okay? So we employ structural biology to see things and how we can have a full control of the enzyme. We can do some manipulation, mutations in particular, which allow us to have not just to study, but of course to have them be applied in industrial processes. Okay? So then only some cases we can come up with new enzyme with unnatural reactions. Reaction that is very much uncommon to any given enzymatic processes. So enzyme is a big market for me, for our case. So I'm sure that some of the groups in applied microbiology research centers as well pursuing work about enzymes. So structural biology has been and will be, of course, a wonderful tool to help and give us ideas about how enzymes and protein interacts and so forth. Okay. And of course, the techniques nowadays, techniques has been X-ray crystallography, for example, and other techniques that I've listed over here has been around for some time. And X-rays, the approach of using X-ray crystallography is one tool that not so much exclusive in many laboratories, but of course has been around for again since it was involvement in 1914 and until now. So there's been the methods nowadays for structural biology, protein structure determinations. But if you look, of course, electron microscopy has been around for some time as well. But when Richard Anderson, Nobel laureate, able to design and improvise the cryo-M, the electron microscopy to add in the cryo preservation aspect. So we are able to see atoms. We are able to see molecules using electron microscopy. So this has been revelations, and for the past 10 to 15 years, more and more structure has been obtained with, not to say with ease, but of course, with at least one bottleneck has been removed. In this case, if we are using X-ray crystallography, we must get crystals. Growing crystal is one of, well, it's an art, so it is sometimes it's some kind of a 30% success rate just to get a crystal of any given protein to be crystallized. NMR, of course, has been around, nuclear resonance, utilizing the protein of interest in solution. Okay, so but owing to its technical difficulties and analyzing the data set, the outputs of the NMR spectrum may have some may slow down a bit the processes. But of course, with nowadays we are talking about integration of many tools. Okay, so of course the computational approach and in silico, in other words, also helps to what I call expedite the understanding and to help to integrate all these data sets involved from many facilities, for example, X-rays, cryo-EM, and of course the NMR. Of course, I don't want to go much about this one. I think this is very basic from basic amino acids to understanding proteins, peptides, and protein. Of course, looking into the structural aspect of protein in particular. Okay, so in this case, more and more complex detail of atomic protein structures in terms of protein folding and the assemblies should be able to be obtained. Okay, so and again, very important is aspect that once we have obtained given protein structures, what will be the thing to be extracted, what will be the information to be extracted from these given structures. And this, again, we want to have more and more characterization effort to be done after any given structure is being obtained. Okay. Right. So this can either be more biophysical, biochemical characterizations in terms of enzymes. So if it's in proteins, we can look into interaction studies. Okay. So from my brief visit to brain, I think you have all the facilities required to have this dream, inshallah, being fulfilled. Okay. So of course, very important that from the structure, we should be able to know functions and any given mechanisms of any given protein or enzymes. And of course, to a more fundamental aspect, to see how protein originates and how protein families are being diverse. And from there, of course, structure-based drug design has also been coined nowadays. So this is where the drug nowadays, previously it was more onto ligand-based approach to find some compound and get the identifying where will the compound actually binds, for example, for your natural products. If nowadays, now we have a structure, from there we can design a compound from that given structure or improvise the compound, for example, that has been previously reported. Okay, so this is how the drug industry, pharmaceutical industry has invested so much in the use of structural biology. Okay, and expediting the process of drug design and deliveries. Okay. So I think basically in terms of into structures, most of us, including myself, very much interested into the experimental process, okay, into looking into protein structures. So X-rays, mentioned about NMR, and now you have the recent advanced research facilities of the cryo-EMs. These are tools that that at least one, we call it the ultimate structure biology tools that will help pave way to obtaining good protein structures, and with good protein structures, we should be able to see enough details about what is the interatomic interactions of any given protein. And of course, now AI has been around, and we are not going to run away from it. It has been big contribution in terms of delivering protein structures. In the case of the protein folding and protein determination methods like the AlphaFold and many more. Rosetta Fold is coming through the years. So these are tools that allow us to give predictions of protein structures without the need of this particular state-of-the-art facility. But yet again, it is by mere predictions, we will require the experimental data to complement and to support each other. There are always some errors that the AI may introduce, so the experimental structure will help to improvise and make the model even more better quality. Okay, so basically, of course, using light, the concept of most of the structural biology tools are using light. Okay, but of course, light, unseen light that allow us to see things that is very much difficult to see. So of course, using light microscopy allow us to do many types of cellular magnifications, and with electron microscopy, scanning and transmission electron, even giving us some magnification to a certain degree, should be able to see organelles. Back then, now with cryo-EM, should be able to see beyond that. We are talking about looking into structures. Of course, this should be able to support what X-ray crystallography should be able to give us later on. So it is very interesting to see that maybe the data here should be updated. I think as far as the contribution of structural biology to mankind, is a lot of Nobel laureates, Nobel Prize winners has employed or developed. Okay. So tools that to help people and researchers to understand structural biology. Okay, so of course it's been structural biology, interestingly, still remains in one of the most multidisciplinary sciences, right? Sciences, multidisciplinary. This is very important. So it is not always that the biochemists must work alone to achieve any kind of feats, but of course we involve microbiologists, pharmacists to help this the collaborative effort be renewed, right? So we are talking about nowadays multidisciplinary. Later on, in later stage, we are talking about transdisciplinary. Okay, interactions of many multidisciplinary team to achieve one objective. And of course, I'm privileged to meet some of these Nobel Prize winners, and if you look at the list over here, so numbers about 29 prizes has been given to at least people that have been involved in structural biology. So hope later we may have some people from Southeast Asia or Indonesian people to be, of course, inshallah, Nobel Prize winners soon.
Amen.
Maybe not for me lah, I'm too old. Okay. So this is what structural biology. If we are looking to aspects of looking into protein structures, because of the nature of proteins are very complex. So from the combination of naturally occurring 20 amino acids give way to many shapes, many sizes, many types of folding of protein structures. In this case, for example, is a types of very unique, peculiar folded protein dominated state. This is called antifreeze protein. Right? So protein that typically stops the ice from forming. Okay, so we try to know what makes organism living in the cold region survive. So this is one of the adaptations, okay? Stopping the water from forming ice and from stopping the ice from killing them. Okay, so this is what at least this protein. And from structural biology, we should be able to see this nice assortment of these residues to which interact with water. Okay, so this is what we could not see until we have the structure. Then only be able to we have some understanding how this Pseudomonas, for example, able to survive the freezing temperature. Okay, this is one example. Okay, so from at least nowadays we have the whole genome sequencing process, so that even makes the understanding even faster. Okay, so more and more new proteins are emerging, and of course the term called hypothetical protein with unknown function are emerging from genome sequence. So this allows big opportunity for all of us to explore even more. Okay, and inshallah not just to come up with good publications, of course later on to come up with good product for eventually to serve the community to the end. Okay. All right. So, and of course, structural biology has also at least one of the aspects is the important structures of ribosome. We know ribosome very well. Protein machine, protein factory in the cells that produces a factory in the cell that produces protein. So this one, for example, with the use of some structural biology, crystallography, and microscopy back then. Okay, and of course now with the use of X-rays, people are able to see how ribosome looks like. Of course, we can see them by electron microscopy, but how is what makes the architecture of ribosomes unique because of this presence of the RNAs and the protein called ribosomal protein as well. And of course, you get a protein if you have such open, very hot topic over here. So you can inshallah get the work we published in high impact journals. Okay, this one in particular was published in Science, and of course the Nobel Prize was announced back then in 2009. Award being shared by three winners from Venki Ramakrishnan. Another one is Thomas Tice and Adayonath. And I'm able to see him Venki Ramakrishnan. This was way back this was in Melbourne. We had a talk, we had a discussion. So he's a very difficult person looking into this ribosome and how ribosome work. And now ribosome has been changed by pharmaceutical company because ribosome is very much important part for important drug target. Okay. Another example of using structural biology is just how people are able to find some new things, new discovery from the use of structural biology. For example, this one over here from Tanaka back in 2009, from a group from the Institute of Protein Research, able to find some peculiar shapes of proteins they call it as red river fault structures. This is electron microscopy images. Okay, looking into like a closed vault conformation, and if you can see them on your right, the open vault, meaning that the vault is very much being destroyed and opened up into 3 petals. So they want to know more what does this vault do? What does it? Why is it there in the red ribbon? So of course they use the crystallography. So this is a protein that formed the question mark I showed earlier. One of the building blocks of these proteins is making up this huge domain over here, and this is huge. This is 13 megalodon protein, right? And the interior part is actually empty, hollow, and that paved way to more and more questions to be asked. Okay, so how do we deal with that? Because this is where more translation, cellular studies are to be employed. Okay, so this is ribosome over there in the middle. So you can see the size. Ribosome was big, quite a massive molecule. Now we have found this red river vault that's even larger. So of course with cryo-EM nowadays, I'm sure more and more detail can be observed because cryo-EM should have no issue with dealing with this particular molecules. So this ribosome, this is what how actually structural biology has been opened up many eyes of researchers and scientists to understand many aspects of processes. So how long do I have? Okay. All right. So now modern structural biology nowadays for the past 10 to 20 years is not just about crystallography. So nowadays people have been employing many methods to actually get more information because more and more wealth information is obtained, the more question is being asked. So, and of course, this data management thereby comes the data science field that's been developed nowadays, just to, of course, help bioinformaticians to manage this, this plethora of research data. So people have been using, of course, X-ray and lasers. Now, cryo-EMs is also not one of the important tools nowadays, other than nuclear magnetic resonance. And of course, other, I would say, cheaper alternative, the use of circular dichroism, spectropolarimeters, fluorescence spectroscopy is very much quite common nowadays. Light scattering tools, dynamic light scattering is very much to see this particle size and the the viscosity of certain samples, and not to forget computational molecular modelings and simulations are also being employed to complement all these methods nowadays. And new things being used nowadays to so-called advance further the use of mass spec, which is the cross-linking protein. Cross-link protein structures using mass spec. So this is quite new, but and again, it is again as what we know before from mass spec. It gives us a detail of protein fragments. So whenever it is cross-linked, we should be able to form and create that Lego set and build up eventually building up protein structures with the help of all these other tools. Okay. And very important nowadays, these tools, understanding of structural biology typically employs the use of recombinant proteins. In some of the case, the protein will require in huge quantity, in milligrams per mil of protein. So this will require protein to be produced in recombinant hosts, heterologously, in E. coli, Bacillus, Pseudomonas, mammalian cells, and many more. Well, of course, we can use protein that comes naturally from the natural resources, but again, we have again to figure out how do we get the enough quantity of that given protein that is going to be studied. Okay, so just from here onwards, just share a bit about how we use structural biology in our case, and some examples. Hopefully, you'll be able to give you some ideas about the plans and maybe some hopefully future collaborations in these particular fields. So this is one of the earliest crystal, the first crystal in Malaysia. So protein crystal was initiated. The project was initiated back in 2004. So this is very much a crystal of a lattice. Okay? So protein crystals, precursor, very much important. Important aspect if we want to employ crystallography, X-ray crystallography. We will require a crystal. So this is the great bottleneck when it comes to the use of these particular methods. We must get the protein to grow as a crystal. Okay, so without the crystal, it's just going to be like a daydream. We are not going to get much of information. Okay, so right, so this is small, very small. Talking about 0.4 to 0.5 millimeters in size. Okay, so that's where we're going to use a lot of some microscopy to handle the small crystals, and why we grow this crystal of size, because some of the X-ray source, the beam size can be very big, and if it's not suitable for the crystal size, it will damage the crystal, and thereby you're not getting much data. Okay, so this is a structure actually coming from these particular crystals, and yeah, so of course from these crystals, this lipase actually comes from our own backyard, from our own palm oil mill effluents, from Bacillus. So from this, the structures have given us a lot of understanding about how this lipase work, and thereby we can do this in many types of processes. Like in the case of producing fatty acids, producing esters for biodiesel, producing certain esters for cosmetic processes, for lipsticks, for any case of pharmaceutical drugs. So this from the structure, we should be able to use the enzyme in many what we call this unnatural processes, unnatural reactions. We can, it's not enough, instead of putting the enzyme, reacting the enzyme in the presence of buffer or water, we can put them in organic solvents, for example. Okay, so but how the lipase survive in organic solvent will require this understanding of in terms of structural aspect. Right, so basically this has been one of the key enzymes, one of the earliest, and a lot of studies has been conducted. And yeah, so that's again me. There's the person in the middle is our first astronaut. It's me back in 2009 when I did my PhD. So this crystal was launched, the protein was launched to space. so to the International Space Station. So to understand how the protein was actually solved, how the crystal is grown in the zero microgravity. Okay, so that's one of the key target, and not just to grow crystal, but from there, how we should be able to see how protein behave in a zero gravity environment. So the person in the middle went to space, but not the one on the left. Not on the left. Okay, so some of the what astronauts perform. Okay, some of the this was back in 2007. Our first astronaut performed this microgravity environments for 8 days, and we are able to get some good crystals from there. So some of the examples that when of course some of the things that we can do is, of course, nowadays not just sending crystal to space is just an opportunity. It's not a must. Okay, so if we have the opportunity comes to us, then we send them. Okay, we send them. But then we had this one. So back in 2007 and 2009 and 2013, we had collaboration with Japanese space exploration agencies to more missions to space. With 6 missions, we are able to send about 34 proteins. We are able to get some 18 structures, okay, being solved from this particular project. Okay. So beyond that, other aspects of structural biology that we utilize that hopefully to help to give some idea about how we use structural biology is, of course, in this case, to understand a protein called aquaporin. So this is a water channel protein that exclusively transport water from in and outside the cells. So in this particular case, this is a highly hydrophobic protein because it only resides in the membrane. And of course, this protein is in the form of a tetramer. You can see this the color coding over here represents 4 subunits forming this form of tetrameric assemblies. So each of this pole over here is where the water passes through. So we want to know, in this case, this is in Antarctic Pseudomonas. So these Pseudomonas are able to survive, for example, at low temperature, and how water is being transported at that particular low temperature. So from this particular case, we want to see how this protein select water and filter certain solutes in order to get the water in and out of the process. So we employ computational approach, and of course, because this is very much a difficult protein to be crystallized, and hopefully we try to get the structures using cryo-EM, because cryo-EM is one of the best thing is that at this moment you do not need the protein to be crystallized. Okay, it can be done in a solution. So this is part of the simulation what the computational should be able to give us. Okay, so computational biology, so very much a subfield of structural biology. So it helps us to understand many aspects of biological processes using computational approach. So here, for example, we are able to see how the gating process of that tunnel. So how the protein perform like the gating stops the ice or water from going out the cells. So this is one way of preserving the water in the cells. So we quite fascinated to see this actually. So, and of course we want to see, this is of course the predicted model using the AlphaFold, using the AI tools, and from there we want to complement with the structural biology in terms of experimental techniques to determine the structures. So hopefully in the coming years, we hope to complement this information and more understanding can be obtained. Okay, another example of our case is this carboxylate acid reductase. This is an enzyme that is belonging to, of course, the EC1 to 130 that catalyzes the conversion of so-called NADP and ADPase dependent reactions from acid to aldehyde. Okay, very important that this enzyme is important in biofuel processing. So because it can convert from fatty acids to aldehyde. Aldehyde is a precursor to many forms of biofuels because acid is bad to have in any hydrocarbon processing. So we want to reduce the amount of acid, so we try to convert them to aldehyde biocatalytically, okay, using biochemical processes. So This case, we have enzymes coming from the Mycobacterium species. So we, at this point, a lot of few groups have been studying this enzyme as well, but only that the productions and conversion of aldehyde is very poor. So, so how do we improve them? Of course, by structural biology, we should be able to understand this how CAR, for example, looks like, and then we should be able to activate CAR. So we are here, of course, we want to know how we can improve and productions of aldehyde, and this is very much precursor to many other products like alcohol, amines, alkene, and esters, and very much useful. And because that's the target, we want to know on what purpose, why do we want to study CAR? Because to help this improvement of these renewable energy processes. So little is known about this. So we propose that the activation is enzyme activation is lacking. So what we want to propose in this particular work is, of course, we want to add another enzyme called phosphopentenyl transferase. So this enzyme is very much important to activate CAR. So, but we want to know where this PPK, for example, binds to this particular CAR protein. So we are able to succeed. We have succeeded in determining the processes, and this has allowed this CAR to be more activated and more aldehyde able to be produced. Okay? And of course, we also employed some of these. Employed some of the computational processes just to help us see and how is the CAR processes in producing aldehyde at certain temperatures. So this we utilizes a computational approach plus this structures coming from crystallography as well to give us some idea about how we can improvise this enzyme to work better and at least the near industrial processes to produce biofuel. Okay, I think this shall be, I think, one final example of my how we employ structural biology is the understanding of very much again cyclophilic enzymes. These are enzymes that are cold-loving, like to work in the cold, and organic solvent tolerant enzymes. The enzymes that are able to work in this so-called organic solvent like hexane, benzene, toluene, so which is unnatural to any given enzyme. So we understand very well that in order for any given enzyme to be functioning, to have some certain activity, so more flexibility and stability of enzyme needs to be hidden and needs to be studied. So this actually allows any given enzyme to be operating at extreme condition. If the enzyme is stable, it is very much heat stable. Less stable, meaning that the enzyme will not be able to operate high temperature. So they typically have more flexible conformation, more flexible shape. One example is our case from Lapis again, from Pseudomonas. So this is from this structural biology, we are able to get good information from at least this is earlier structure, because again the bottleneck of crystallography, we were not able to get the crystal structure. It's very difficult. That's the 50%. I am in this 70%. I'm just a cool person. So but then of course it's not the end of the study. We utilize other tools to help us understand and help us to obtain this structure of this particular lattice. So lattice has a non-catalytic domain, catalytic domain as well, where the reaction happens. And from there, of course, we proceed further and to remember that I was not able to get the structure obtained. So we use other types of processes. You call small angle X-ray scattering. Another tools, again, this is a collaborative efforts from my team and with a group in Thailand. Thailand light, we call this Siam light source. So they have, of course, thankfully they have been able to provide some facilities for us to able to look into the protein structures in solution. So again, here we are able to find how the protein moves in the solution, and we are able to get this number of structures obtained and to complement with the computational data as well. Okay, so then the number, this is so I call this ID for the database for small angle data. So we have protein data bank. We do have also this small angle scattering data bank, so the number is SASDEX9. Okay, so this is what we see. Okay, over there. So what we see in the structures in solution, so the protein behave and form these types of conformation. It moves in solution. So when this happens, so crystallization may be difficult. So this explains why we are not able to get the crystals, because in the ununiformity of this protein shape in the solution. Okay, so but then we have other tools. Okay, now for example, now cryo-EM should be able to give us detailed structural aspect of this particular protein. Okay, probably I just skip to this one. Okay. Okay. Okay, briefly about this particular protein and enzyme. This lipase has a lead region. So lead is the region where it protects the active site. So, and we try to want to know this, whether this lead region is important for the substrate specificity of the enzyme or the solvent entry of this particular enzyme. So we perform some molecular simulations, and this particular region is in this particular lead 2 is moving rapidly up and down. So this tells us that the structure, the lead region is opening in an active form. So in the presence, bear in mind, this is in the presence of organic solvent. So with this, we have some early indicator about how the enzymes are able to work in organic solvents and how it selects different substrate in order to perform many types of reactions. Okay, some of the routine, I think some of these comparison of structures before and after. So remember, the lead 2 is the one that gives and open up the cavity of the active sites and to allow the entry of the enzymes, allow the entry of these substrates. So we are actually looking at the top view. So this is actually the top part of the active sites. When it is closed, okay, when it is open, it gives us some tunnels. So substrate tunneling allows us to explain the substrate entries and product release, or even the entry of any given molecules into that particular active site cavities. So this is not seen to other lipases, as far as I'm concerned. Okay, so what the solvent does to the enzyme is that it actually pulls the active site even wider. So the active site interacts with the solvent, and the solvent actually sort of enlarge the enzyme cavity, substrate active site cavity. So this again helps us to explain why the broad substrate preference for this particular enzyme. So without the structure, we may not be able to explain so much. It's just that we know from the in vitro analysis, but in silico, this is what actually the structure is giving us in the front. Okay, so from there, of course, we want to know from this understanding of organic solvent or the reactions of enzyme in organic solvents. We try to put up, can this enzyme produce some aromatic flavors? Lipases, this enzyme can produce strawberry smell, durian smell, and with the interactions of many types of esters. Okay, so these aromatic flavors again can now be produced enzymatically, okay, and more rapidly, by the use of enzyme. So with this, we can avoid the use of harsh chemical, high reaction processes. Okay, so some of the example, I think, don't go much about this one. So here we are able to prove that this enzyme is from structural biology. This enzyme are able to give us a lot of understanding, not just in terms of later on the output is not just in terms of publication. We are able to have this one patented, and industry should be able to take this technology and be produced to have this particular esters be produced in their formulation, cosmetics, pharmaceutical processes, and so on. Okay, so just to in summary, I think that of course some of the great crystals obtained in our lab, again, sooner or later, some of you should be going to this field. This has shed wonderful lights, wonderful findings, unseen, okay, by our naked eyes. So of course, again. So, but with these structural biology tools, okay, like the cryo-EMs, the X-rays, okay, you have in this campus, in these Kibidong areas, and of course, some of the tools employing in this synchrotron facilities. Okay, not many in the world, but of course, available out there. So we should be able to have more and more detailed understanding and to advance further about this our research in the modern biology, right? So I guess that's all from me, I suppose. Thank you. Thanks for listening. That's our team, basically, some of our students and co-PIs over there, just completing our 1 hour annual bowling event. Okay, so yeah, that's all. Thank you. Thank you for listening. Good.
Thank you very much for your inspiring presentation, Prof. Sukuri. So for all of the audience, both in offline or online, now that is a Q&A session. So all of the PQs that join our session today can discuss all about the structural biology that already conveyed by Prof. Sukuri today. So I'd like first to give the chance to the audience in the offline session. Is there anyone who would like to discuss with Prof. Sukuri here?
Yeah, okay.
Please.
Thank you for all the presentations, Professor. It's really interesting. So actually, I'm not a chemist, so I'm a zoologist and approach with many animals. So from insects through mammals. So what I'm asking is that it is really interesting because the biostructures, proteins may cover from many organisms, also especially in the sensitive animals. So what I'm asking is, to what extent the purifications of the proteins that will be used in the final electron microscope analysis should be done? I mean, should be really pure protein? Because, for example, we extracted the keratin from the chromophile, and we want to see the structures of the keratin. But in chromophile, they have like fat and then another protein from the bacteria or from another organisms, and also the purification is required. So, to what extent purification should be done before we analyze these structures of the keratins in the microbiome?
Slowly, okay. Okay. So basically, structure from cryo-EM, I have little experience on that actually. But basically, in terms of structural biology, the use of protein for any given structural biology approach typically must come with a very pure protein. Okay, we are talking about 99% pure. Okay, yes, very pure. Because in terms of structural biology determinations, we wish to eliminate as much impurities as possible. Because in crystallography, for example, this purity is important when it comes to forming some good crystals. So if it's impure 95%, even the impurities may come from the protein itself. Over time, the protein may experience some degradations. So these small fragments may, of course, interfere with the crystallization process, and eventually no structures are okay. I believe that in cryo-EM, so I think you can actually sacrifice a bit on some purities, I suppose. Is it true? So Dr. Fina probably have more detailed answers of that. But to analyze some so-called single particle, this homogeneity and of a certain protein is very important. But again, a good question is that by the way. So we must ensure the homogeneity of the protein. Because some proteins either comes as a so-called subunit, multiple subunit. So in solution or in actual environment, what is the nature of this protein assembly? Okay, so it's either the protein may be in multiple oligomers, but how do we maintain them? Okay, so we need to have this protein to be very homogeneous, not to have mixture of monomer or dimers or tetramers. So this typically calls for some good buffer preparations, and and of course it should be very much pure. Or before we try to troubleshoot the cause of some formation of other impurities. Yeah, that's what I can actually add up to that. I hope, probably Dr. Fina. Yeah.
Mungkin menambahkan sedikit ya dengan Pak siapa? Oh ya, dengan Pak Bramantio. Jadi memang kalau sebenarnya sesuai dengan yang diungkapkan Prof. Supuri tadi, intinya kalau untuk mendapatkan 3D structure protein itu kita memang membutuhkan proteinnya dalam bentuk sangat pure, karena untuk prinsip di cryo-EM sendiri ini sebenarnya prinsipnya adalah kita mengambil gambar 2 dimensi dari berbagai sisi, dari berbagai angle gitu ya. Nah, kita bayangkan kalau nanti ternyata sampel dari Bapak Ibu di sini ini ternyata tidak pure, misalkan ada Mas Bramantio dengan Mas yang di sebelahnya ini saya foto dari berbagai sisi, kemudian dengan menggunakan AI, mereka ini akan nanti akan digabungkan seperti kita menyusun puzzle gitu ya. Jadi kalau ternyata ada 2 orang nanti digabung jadi satu, nanti jadi berbeda nanti hasil struktur 3 dimensinya. Jadi emang sangat penting untuk proteinnya itu bisa setinggi mungkin tingkat purity-nya ya, tingkat keurniannya. Nah ini bagaimana caranya? Sebenarnya akan lebih baik kalau protein Bapak Ibu ini diproduksi dengan teknologi kombinan. Jadi nanti proses purifikasinya akan lebih mudah, walaupun kalaupun dipurifikasi dari sumber aslinya juga bisa, tapi seperti yang sudah Prof sampaikan tadi di presentasinya ya, untuk mendapatkan kuantiti jumlah dalam bentuk pure-nya ini akan effortnya akan lebih tinggi. Jadi mungkin itu saran saya.
I have experienced to work with EM. It may be a little bit the same with the cryo, but the difference in some situation, I will need to read a small grid, and then I put samples on the grid. And TM is really flexible because you cannot find what you want in the grids that you put inside the TM. You can just put it out, and then you can go to the microsome, and then you have new slides, and then you have new sections that we want to observe. Is it really flexible with cryo, and we want to, for example, the first analysis was not really pure, and then we redo the analysis and going somewhere else, and then we go to the same place again to do the analysis. Is this the right idea? Or we need to really whole things from the first.
Okay, actually in cryo-EM has like 3 methods that we use. But for the 3D structure protein, we use either single particle analysis or microelectron diffraction. In the single particle analysis, we use the protein in liquid form, usually in the pure form, with there is like a ranging of the concentration of protein as well that need there is like a special qualification as well. But in the microelectron diffraction, it's like a little bit similar with X-ray crystallography. They use crystal. So the choice is between either using single particle analysis or microelectron diffraction. In the single particle analysis, as the name suppose, it needs to be single. So single gain needs to be in pure form. Yes. So whatever technique that you do must be provided the sample in the purity form. Yes. So that's why I offer you like I use the recombinant DNA technique because using that you use another host so you can produce in much higher than in the natural way because it is like engineered one. Yes. Yeah, maybe Prof
Yeah,
can.
maybe I can. I think quantity, and because in terms of doing dealing with some structural biology aspect, protein typically require quality and quantity. Quality means that the protein is pure, and getting a pure protein is one thing, getting a high quantity of protein is another thing. So this is, I think, if there's opportunity for you to actually use recombinant techniques, please do so. That is, I think, advisable. Most of the techniques require a huge amount of, not huge amount of protein. To this will typically use the use of E. coli, for example. But if Bacillus, Pseudomonas species, and even more complex cells like yeast as host and even mammalian cells. So so this again will facilitate, recombinant protein will facilitate, helps us to improvise the protein application, and eventually with the advent of chromatographic techniques nowadays, they should be able to help you to get enough protein suitable for structural biology purpose.
Basically, so I will underline that believing is exponential analysis should be done with the recommended. I mean, if we do with the OTX, the natural resource extraction, it will be this is not before
Yeah, so it depends. If you are able to achieve, you're able to get some enough quantity, milligrams protein, then
quality and
yeah, quantity. Yeah.
Okay.
Yep.
Okay,
Right. Thank you.
Okay, thank you. I would like to give chance to other participants in this room. Or I will move first to the our colleagues in the online sessions. Already? Oh yeah, okay.
Thank you. And critical and the active side of the plan to know the so what's the to equipment last week crystallization first or the AM or and throughout the language and we said that is what the line.
Okay, thank you. That's a difficult question for me to answer. Okay, right. So it is an interesting question, by the way. So X-ray crystallographies and cryo-EM both eventually in the end serve different similar purpose, which is to provide us some three-dimensional structures of protein or enzymes in particular. So crystallography, X-ray crystallography, protein crystallization, those are from similar terms actually. So X-ray crystallography, we will require a protein to be crystallized. Okay, protein, that is the processes, we call it as protein crystallization. And the crystal is then to be used for the X-ray light, then we eventually get some structures. But the protein has to be in the form of crystals. Okay, so as I presented earlier, my experience with my lipases and other proteins. So we have to crystallize the protein, and thereby we have to employ this under an X-ray light, for example, and X-ray crystallography. So cryo-EM will of course eventually provide similar data, for example, similar information in terms of, for example, you want to see some active site. So only that it does not require any crystals. So it requires a protein to be in solutions. Okay, but yet again, the term cryo-EM is that the sample has to be, to be frozen and it's under what we call under very low temperatures, and that yet again under certain electron what we call magnification, we should be able to get some structures as well. Only that the source of the what we call this how the form of the protein is either to be in a crystal state or in solution, for example. So crystallography typically must have to be in crystallized state. And again, this will give you information. So certain information about enzyme and active site, we should be able to see them from the use of these both tools actually. And there are of course advantages and disadvantages. Briefly for crystallography is that the size of the, for example, if you want to see a protein, for example, of your interest, the size is unlimited. So you can have this protein from small size to medium size, large size. So, and it is not a big issue, but only that you need to have the crystal to be crystallized, the protein to be crystallized. This is the bottleneck that I mentioned earlier. It can either be 30%, 40% success rate. Not all protein can be crystallized. And for the so-called cryo-EM, I'm sure that they have some limitations in terms of size, but that is not called a stumbling block as well. But I'm sure that in coming years, the more and more smaller structures, I hope, can be obtained. But the issue is that protein with at least a minimum of 100 kilodalton or even larger is required if you want to employ cryo-EMs. So crystallography, so it's not an issue. You the less than 10 or 15 or even to the large extent of ribosome just now and megalodon size, yeah, you can use that. So some of this, well, at least brief comparison between those two approaches. Hope you get some use.
So I think the sample is for analysis or GB might be during crystallization.
Okay, yeah.
Bring to Indonesia or time, so can we for cryo-EM?
cryo-EM over here?
Is the sample from Malaysia?
Uh-huh.
And then
Oh, yes, I'm sure, yes. Yeah, sure, can. Possible. In the protein in solution, for example. Yeah, can be shipped. Yeah. And analysis can be done over here. Is that the question?
that must be phrased in this.
Must be in freezing state. Oh yeah, so depending on the shipping, yeah, some of which I think the proteins best to be in a frozen state. Yeah, call it. This is in the case if you want to have a sample be shipped from Malaysia and then be analyzed in Indonesia. Yeah, so yeah, that is possible. Yeah, so we have done in our case, so we ship protein, for example, and from Malaysia to Japan. So it can be done, and yeah, our counterpart in Japan doing all the analysis, data collections. Yeah. Yeah. Thank you. I hope I answered your question. Yeah.
Thank you.
Yeah. Sorry. Yeah. Okay.
Okay, thank you Bu Ira. Sangat bisa ya harusnya kalau dari luar. Dan tipe pengirimannya itu tergantung dari karakteristik proteinnya. Kalau memang proteinnya stabil, termostat stabil di suhu tinggi misalkan, ya mungkin tidak perlu pengiriman khusus ya. Tapi kalau untuk protein-protein yang sensitif, misalkan dia memang harus di suhu tertentu, berarti nanti tipe pengirimannya juga mengikuti. Jadi intinya tergantung dari karakteristik protein yang akan dianalisakan. Seperti itu kira-kira ya. Thank you Bu Ira. Then maybe I can move to the our colleague in the online session. If there anyone who want to ask questions, can directly open the mute, or maybe can raise hands, or can also write your question in the chat box. Then I will convey your question to the prof in here. Okay, well, waiting. Maybe is there any question as well in our colleague in the offline room? Okay, please.
Yeah.
Thank you. Thank you, Professor Shukri. I'm from the ecology. When you said talk about found about the microorganism in the Arctic and in the
Atlantic, yeah. South Pole.
Atlantic, they have some protein they can unfreeze. And that's why I think, okay, maybe in our research, we can apply to the this methodologies research. So knew about what happened with the organism like a plant or animal. They can adaptation with the environment. And I just think about for the macroorganism, what can we do to take the sample? Because from our area, they said we can set up to know what the life is working from the cell, and then the cell from the wood, they can produce the protein from the wood. I just flashback from. That's why how the mechanism, the life working in our work for the environment. Because I just think like that. I'm the aquatic researcher, and I get a phenomenon like the shark. They have oil, and the oil from the heart. In the local people used for the medicine. I just think maybe from the where is the compartment from the liver produce the oil, and then link between the organism, the producent, the protein or something, become the research like the old method from the organism, and then we can get knowledge from the working the system.
Thank you. If I understand the question correctly, how do we, for example, Kachibali can link with that particular phenomena? How, for example, production of certain compound, okay, some lipid, in certain in terms of cellular processes. Okay, so I think a lot to be explored from there. I think that is by the information from, I think typically for a start, if you have enough bioinformatics data, for example, then at least should give us some really idea about what proteins actually involved in that pathway actually. So the pathways of producing that particular lipid. Then how, for example, the lipid is being translocated, is being packaged. For example, typically that will involve some protein as well. Okay. So I think from there, I think it's not going to be like a one protein involvement. So it will involve many types of transporter protein, transporter and enzyme as well, because biosynthetic pathways for lipid is big, it's huge. So unless we want to see the key enzymes that is important towards the so-called the production of that lipid, So that key enzyme can be determined can be estimated from bioinformatic analysis. For example, if we have some genome database, so we can predict from pathways from what we call this CAG pathways. So criteria for genome and genes and genome. So from there, we should be able to map out certain pathways that are able to in producing that kind of lipid. So then I think in terms of transportation from there, so we should be able to at least find some of these we call the key enzymes. Okay, some of which can be enzymes that involve in the feedback inhibition, for example, that may be triggered by certain hormones, certain proteins that triggered, meaning that the enzymes are activated to produce lipid along the way. So I think that is very important. I think to at least the fundamental part, and how the lipid is being used by the cellular at the cellular level is another level. I think I would go to that aspect on the probably that lipid may be unique to that particular aquatic organisms, for example. Yeah, so that's what I can suggest. Going to the genome sequence and the bioinformatics. When we have the so-called the target protein or target enzyme, and that's where the structural biology starts. So where we want to see how the enzyme interact, how the enzyme produces such lipid, maybe through going through some metabolic pathways of beta oxidation, maybe. So this is where the structural biology will come and get the structures, and probably a new structures, probably new enzymes. Yeah, that's my brief. I hope.
Very complex world, we can get information like how the life working.
Yes, yeah, very complex. But I think that we have some precursors. The initial preliminary ideas, preliminary attempts is there. So from there you can expand. Once you have at least know how the lipid, for example, is being produced, then only how the lipid is being used. That's another big project. Thank you.
Thank you.
Okay. Thank you dengan Bu siapa?
Iya.
Oh ya, thank you Bu Dian for the questions. I hope it helps. And then chance to everyone join our discussion today. Is there anyone? Okay, please. Waalaikumsalam.
Saya ingin bertanya bentuk dan struktur kita dari materi. Misalnya mengajarkan bagaimana kita sumber lipid pada beberapa organisme frekuensi dan bagaimana respon perubahan dari enzim fase tadi. Jadi pertanyaan seperti Pak sampaikan ketika akan menggunakan fase yang memang spesifik fase program jenis apakah memang